Hello everyone, I'm Yash Sondhi, and today, as part of the symposium on open source tools, I'm going to tell you about the work we did to develop an open source and portable solution to monitoring animal locomotion and how we benchmarked it in the lab and the field. This work was done at Florida National University under Jamie Theobald and in collaboration with Dr. Akito Kawahara and Matt DeGennaro. So, as humans, circadian rhythms play a major role in our lives. We've all experienced jet lag, and on the top left is an example of what are the key driving factors, say food and light, that can entrain our circadian rhythm after, say, a jet lag or something disrupts it. Um, Many of us work better in the morning, some of us work better at night, and to the right, you see a graph of different chronotypes and how some people are genetically predisposed to sleep and wake up at slightly different times. And the third bit is a lot of our physiological functions are also linked to circadian clocks, not just being sleep and awake. For example, our body temperature also fluctuates with a 24-hour cycle. As we get closer to the night, we cool down and we sort of warm up as we get closer to the day. Now, circadian control is not just in humans, it's exhibited across the entire animal kingdom. Many behaviors like reproduction, feeding, eclosion, and flight are under circadian control. And this is true not just in animals that have a brain, but also in animals without a central nervous system. There's this really cool research where they found jellyfish also exhibit sleep and wake cycles. And even behaviors like coral spawning can be under circadian control. And plants exhibit too. This is an example of a plant growing that was filmed over a course of 2 weeks. And you can see on the right where all that's been plotted is the average pixel motion, or the number of pixels that have changed across each image. You can see that it fluctuates with a circadian rhythm. Now, insects were key models to uncovering both the physiology and some of the genetics behind circadian rhythms, and work done in bees, flies, and moths has been super important. Carl von Frisch uncovered some of the waggle dance in bees, and what fewer people know is that bees use the sun to navigate to those locations, but they can also compensate for the sun as it moves. And they do that only because they have a circadian clock. Similarly, some of the classic genetic experiments by Seymour Benzer in 1970s and his colleagues were able to identify the mutants with different DL cycles and were able to go back and figure out which genes were involved in some of the circadian control. And they found they're conserved across many, many species of animals. And moths are being studied for pheromone release, which is under circadian control, which is highly important for integrated pest management, because when you set up a trap, needs to coincide with when the moth actually calls and whether it's active at that time. And I'm going to focus a lot about moths because that's my main thesis topic. But this is an experiment where in the 1970s, they looked at pupil eclosion, which is moths that come out at different times. And they attached a string and a lever to a drum that they attached to a moth at the other end, or a moth pupae. And they monitored this in a light and dark cycle and showed that different moth species eclose at different times. But as you can see, this was an ingenious setup, but it was pretty bulky. And even today, while we can collect a lot more data with infrared beam devices and we reduce the size, the gold standard data for circadian rhythm, the devices that collect it are still really bulky and expensive, and they're mostly for lab use. And one of the disadvantages of doing this in the lab is most people use abrupt light cycles. This graph is circadian activity for two different moth species collected by some colleagues at Cornell. And they've got some really neat data, but they had to manually digitize a lot of this, and they used on-off lights to simulate a light and dark cycle. But in reality, in nature, light cycles are much more gradual, and especially when many moths start becoming active at dusk and dawn, there's almost a 4 or 5 fold decrease in intensity. So this graph shows how, as the sun or moon goes through the sky at different elevations, how illumination changes. And you can see that as you go from, say, just before sunset, start of twilight, to the end of twilight, you get a really sharp drop in intensity over the course of maybe half an hour to a minute, depending on where you are. And similarly, at night when the moon is out. So this really matters to a lot of organisms that are active at this time, are starting to get active. And we wanted to start studying this behavior in a little bit more detail. So to address some of the issues, we came up with a device which is what paper has been recently published or is in press at Methods of Ecology and Evolution. And you can scan the QR code to access the preprint, and we call it the Portable Locomotion Activity Monitor. It's a cost-effective setup for robust activity tracking in small animals. And I must thank the team of people that made this project possible. I didn't believe that anyone else besides me would want to use it, but my advisors and my colleagues pushed me to publish this, and thanks to everyone's effort, this paper is finally through. And now I'm a lot more excited about it because of the positive feedback that I received from a lot of people in the community. And just the whole process of making it was a lot of fun. So let me tell you a little bit about what we came up with. So we used a Raspberry Pi, which is a small microcomputer, an infrared camera, and infrared lights with to capture motion using an open source motion software. It's called Motion. And it captures motion by recording what's in its field of view all the time. And as soon as there's any two frames that have more than a certain level of pixel differences, it starts what it calls a motion event. And once it starts, it can either take pictures, videos, or run a script, and it keeps recording the motion event till it finds that there's no motion for a predefined period of time, say 2 seconds or 5 frames. Now, this device was used for bird monitoring. It's used for a lot of other things, but typically it is used in visible light. And by adding it or using it with an infrared camera and using infrared lights, we can record behavior throughout the clock, even in darkness, which was a huge issue for the kind of experiments we wanted to do. Recording animal motion in the dark is fairly challenging. So let me show you what the device actually looks like. So this is the Raspberry Pi, or the microcomputer. This is the IR camera, and these are two onboard IR lights. When we use it in the field, that's the extra infrared illuminators, and this entire setup can either be run off a power bank and portable software, except maybe the big external lights, or it can be run off mains with an uninterrupted power supply that just makes it more robust in the field. Um, and we run things in mesh cages because we found that limiting the area in which the animal is active is a lot better for collecting more data. And we typically run with multiple individuals, but this can also be run in the open with one or two or as many individuals, because you're capturing pixel difference. As long as the animal motion is visible in the field of view and it's large enough that it can be captured as enough pixels, it gets detected. And to make life easier for everyone, including ourselves, we took the hardware and the software and sort of made it easy to replicate by creating a disk image of the software that can be cloned on different Raspberry Pis without having to reinstall everything each time. And we also added analysis scripts. We added scripts to do memory transfer and to configure it, all in a manner that you can sort of go into the field and set it up really easily. Or if you have access to a lab, and you can do it in the lab too. Um, so the first thing we did was we decided to benchmark a setup, and we used induced motion in beads and then stop the motion. So on the left, you see this is what the graph would look like if it was detecting motion properly, and this is what it should look like when it doesn't detect motion, when there is no motion. But and we did this for 3 different bead sizes: 2 millimeters, 8 millimeters, and 15 meters, with multiple replicates. So each one of these is a replicate. And you can see that for larger beads, this works fairly robustly. But for smaller beads, it starts getting a little patchy. And part of that is that it works better as we got it closer and closer to the lens. But if we were doing this at the same distance, say 20 centimeters, where we'd expect insects to be flying, the data isn't as clean as you get to the limit of resolution of the object. But you can improve this by giving it more light or getting it closer and closer to the field of view of the lens so that it appears larger on the lens and registers as consistent pixel differences. And what that translates to in the lab, I'll tell you. So we then decided to test this device with a commercial activity detector. This is what some of what I'd shown you earlier, and these are called LAMs, which was sort of the inspiration for the name, where they use an infrared beam with multiple tubes. And the idea is you put a fly or an insect in this tube, and every time it passes through this beam, there's a detector set of detectors here, and it breaks the beam and counts it as an activity event. And if you put food on one side, hope typically the flies or moths, whatever insect you're putting, can last for about 4 to 5, maybe 7 days, and you get counts of beam breaking events over the course of the duration, and each one of the channels is separate. So you get data for 30 to 50 flies or whatever insect you're putting in. The disadvantage is these devices are really expensive. They're about $4,000, but they do work in the lab in a controlled setting. um, so what we also added to this is we wanted to get natural light cycles. So we used controllable LEDs. Each one of these LEDs can be controlled by a Raspberry Pi to set brightness and color. And we basically ran a script that controlled the light level to sort of match a dusk and dawn, not just an on off. So you have light levels go from say 75 to 0 lux. This is relative. So numbers don't matter as much as how the light levels drop and how long they drop. This is just with a $5 light sensor that sits on the Raspberry Pi. But the nice part is the light levels and the Pi and the camera, all of it can be controlled by the Pi, and the light measurement too. So it's pretty self-contained. But anyway, we built this for running it in the lab to sort of mimic natural light cycles, mostly because COVID hit and I couldn't go to the field. But doing this in the lab, I realized the advantage of testing some of these in the lab setting, and it worked. So we did this with moths, mosquitoes, and flies. And to the top panel are the pLAMs of our device, and the bottom panel is the data from the LAMs. And you can see that while the actual raw data looks a little different because they are measuring different things. One is measuring beam breaking events, one is measuring motion events. But when we looked at the total number of activity events or motion events or beam breaking events over each hour for the whole experimental duration, we didn't find the significant difference between the two devices. The top panel is basically the pixel difference over each day for each event, or the maximum pixel difference where there was most motion. And in the bottom, it's the number of counts in a minute. Um, so we did this for mosquitoes. Data was not significantly different for mosquitoes. We did this for flies, and the data was a little different. I mean, there were significant differences when we used certain tests. We compared the data using t-tests and Wilcoxon rank-sum tests. So we had to check whether it was normalized or not. And I don't think this is the best way to compare this data, but if anyone has any suggestions, I'd be happy to hear them. It's the best we could come up with. Um, and for fruit flies, we found that because they were smaller, we were able to capture flight behavior really well. And we think that the LAMs actually were capturing more walking behavior because we expect that fruit flies wouldn't fly after dark. And that's sort of what we got with when we looked at a device. And when we looked at the images, we found that it's capturing flight really well. But we found that the LAMs are, because they're in a tube, they're not expressing their full range of behaviors. So you almost get a biased result here, where if you looked at this, you'd find that, oh, they're active at night, but they don't fly at night. And for a lot of vision and questions we're interested in, we're more interested in when they fly. But then we took it to the field, sort of the ultimate test. We somehow managed to get to Costa Rica this summer with with these are my two advisors, my amazing field assistants, and some other lab managers and undergrads who did an amazing job. And we were lucky to get gorgeous moth screens with thousands of moths in the tropics. And yeah, so we went there, we set up 6 LAMs for about 10 days, put 4 to 8 individuals of different species in these devices and tried to measure their activity. It was hectic, but it was a lot of fun. And I should mention that my first PhD chapter was looking at how different visual genes have evolved in with the El niche, so with species that are day flying, night flying, or both. But as we went out to the literature and tried to quantify that, we found there's very little data on quantitative DL niche, or when different butterflies and moths are active. And to the left is the tree of life for butterflies. Everything in yellow is diurnal or day flying, everything in blue is nocturnal or night flying, and everything in green flies either during the day or during the night or both or something in between. And I won't get into the details of the genetics aspect, but what we found is that we didn't think that the bins of nocturnal, diurnal, or both were clearly enough to define this. It felt like this was a very sparse binning, and we felt there was much more to the story. So when we tested this in the field, we were really happy to report that it worked and worked beyond my wildest expectations. So we managed to get about 12 different species in 10 days with 4 to 8 individuals over at least 48 hours. It did require lots of troubleshooting and a lot of tense nights, but eventually we got it to work, and the next iteration was much, much more seamless. And as you can see that uh, there's a pretty large variation in yellow is diurnal, blue is nocturnal, and green is either dusk or dawn active. And just by looking at this, you can see that some species were completely nocturnal, but a lot of the species that we thought were only day flying also were pretty active. There was a peak at dusk and they were active at night. And there was also species that were active at sort of dusk, dawn, and during the day. So clearly, the story is a lot more complicated than we let ourselves believe. It's not just a nocturnal diurnal, and it would make sense if there's food and some of these mimic toxic beetles, or they can escape from they mimic wasps. Then it would make sense that they fly when their models are active because they're well defended. But why would they not fly at night if they can still find mates and food or can see at night? So looking at this, we think that there's a lot more to the story, and we were quite happy that it worked. But yeah, sort of to summarize, we built this device to be open source, portable, and inexpensive. It costs only about $100 to $120 to build one of these devices. And we wanted it to be easy to assemble, rebuild, and sort of work within the field. All the code is Python based, and we set it up so that you can even do all the analysis on the computer in the field. And while we use it to measure circadian rhythm in moths and insects, it can be used for any animal that shows motion. You can use it to test foraging, predation, eclosion, anything that you can isolate in the camera's field of view, and is motion triggered can be recorded. One of my colleagues has been using it to study how mosquitoes home in on heat versus uh, nectar. People have wanted to use it to study how what time different flies started closing from their pupae. There are an infinite number of possibilities you can use it for, and you can modify the lenses that are on this camera to be either wider field of view or limited field of view. And you can even use other sorts of infrared cameras that are built for the Pi, or high speed cameras. There's a lot you can do. um, but yeah, that's mostly my talk. I want to quickly thank the team who made the fieldwork possible in Costa Rica, the Akito's lab and Jamie's lab. And I would also like to thank a lot of my funding sources and many, many, many other people who made this possible. Thank you. And if you have any questions, please free to contact me on Twitter or through the SIGP app.